draw everything, so they've quickly gone through and not really read the question. Remember, you've got to read and make sure, you don't want to waste your time, you've got to make sure that you're only doing what you need to do. In a track situation, you're going to need to do other things. You have to know, learn how to manage your time, because you're going to expect to produce quality, or be expected to produce quality, but in a timely fashion. So that's why I'm going to give you a few minutes. So now, if we have a look at the marking rubric, you can see on the left-hand side you have some criteria that will get you only 1 mark, and that would need improvement. And on the far right you have full marks for drawing. You have a look at some of those things, and some of the things we've already said. It's label, magnification, scale, it's not coloured in, it's not sketched, some annotations, and so on. So that's very handy to have when you start doing drawings. You've got something to aim for. Okay? Now, the next thing we're going to do is we're going to actually now look at our slide of the human vagina. Some of them are actually rats, but most of them are humans. And we're actually going to do a little drawing based on what we see under our microscope. So, what you're going to do though, there's 2 types of biological drawing. One is a zone diagram, and the other type is where you're going to be showing lots of cellular detail. There's a very simple difference between the two. The zone diagram is generally done at low magnification. In your case today, you're going to be using the 10 times objective lens. And in a more detailed or higher magnified drawing is generally done at 40 times objective or greater. Okay? So this shows lots of detail. We're not going to do that today. Basically, we want you to get some practice at working out your scale and transferring that information that you see down your microscope onto a piece of paper. So today, I'm going to get you to do a zone diagram at 10 times objective of the human vagina slide. Now, no one's going to expect that you are histologists and are masters of this piece of tissue. It's your second week. I don't expect you to know all the anatomical details of this piece of tissue. But what you are likely to see, and the rough kind of idea for this zone diagram, is a zone diagram really just shows different zones. So, like that picture on the screen there, you're going to see in your slide some wavy stuff like this. That's pretty much a zone diagram there. The only difference is this top layer is actually dark purple. It's stained up quite a bit darker than the rest of the underlying tissue. And the rest of the underlying tissue is pink in colour. Okay? And just to give you some fancy names to impress your friends, freak them out, I'm going to put a few labels here. The vagina, like the scalp, like the soles of your feet, and a few other spots, have a type of epithelial tissue, and epithelial tissue is found on the surfaces, either internally or externally. The epithelial tissue is what's exposed to the outside world, if you like. So, different types of epithelial tissue serve different functions, and so therefore have a different structure. So, in your gut, you have a type of epithelial tissue called columnar. It's one cell layer thick, and that's used to absorb nutrients in your gut. All the way along your gut, you have this type of epithelial tissue. In other parts of your body, particularly parts that are dealing with abrasive activities, I guess you call them, you need to be able to have a bit of protection. So if you lost a few layers of cells, it doesn't matter. So in the vagina, the slide you're looking at today, even though this just looks like a big slab of tissue at 10 times objective, if we were to crank that up to 40 times, what you would actually see is hundreds, certainly tens of individual flat cells that go like this all the way to the outside. The little baby or the youngest ones are at the bottom, and the old ones are towards the top. So what that means is, if this was on the sole of your feet, for example, if you were to step on a rock or something, you can lose a couple of them, but you're still protected because you've got quite a few cell layers underneath. So this kind of tissue is called stratified, which just means layers, squamous, which just means flat cells, so it just means lots of layers of flat cells on top of each other. So, when you go to do your drawing of the vagina, you're going to do basically two kind of sections or two different zones of tissue. That's all. You don't need to do the details like you're doing, say for example, your blood, so a little smear there that you just did a little couple of blood cells. You're not going to be showing that level of detail. But what you might see though is you might see some blob thing there. You can draw that. That might be some other type of tissue. It might be a blood vessel. Or you might see some big squiggly thing here. You can draw that. Don't then end up drawing this and then that and this one here and then that. We don't want any details, no cellular details. It's just capturing big, different types of tissue.